The biology of confidence, what your nervous system, gut, hormones, and ancestors know about you that your mind never will. Chapter 1. The Confidence Lie. Why everything you were told about confidence was working at the wrong level. In 2009, a researcher named Dr. Elena Marsh arrived at Harvard Medical School with a question that no one in her department thought was worth asking. She wanted to know what was actually happening inside the bodies of people the world called confident. Not what they were thinking, not what they were telling themselves, not what morning routines they had adopted or which visualization practices they swore by, but what was measurably, physiologically different about them at the level of tissue, nerve, and hormone. She designed a 6-year study. She measured heart rate variability, cortisol awakening response, inflammatory cytokine profiles, vagal tone, gut microbiome diversity, and interoceptive accuracy. She recruited participants across a wide spectrum of self-reported and observer-rated confidence and tracked them across high-stakes performance contexts, job interviews, public presentations, negotiations, first social encounters with strangers. What she found, when the data finally resolved into something coherent enough to publish, was not what the self-help industry wanted to hear. The confident people were not thinking differently. They were not using better self-talk. Their mindsets, as measured by validated psychological instruments, were not significantly more positive than those of their anxious counterparts. What was different was their biology, measurably, consistently, reproducibly different across every physiological marker she had chosen. And the more she looked at the data, the more obvious it became that the thinking had not produced the biology. The biology had produced the thinking. This is a fact that the entire confidence genre has been organized for decades to help you avoid, not out of malice, but out of ignorance. The comfortable, lucrative ignorance of an industry built on the premise that the mind is the source of human behavior rather than its most visible symptom. When a confidence coach tells you to stand taller and believe in yourself, they are not wrong that tall-standing, self-believing people tend to perform better. They are wrong about the direction of causality, and that error, that small, seemingly innocent reversal cause and effect, is the reason you have read the books, done the work, adopted the practices, and still found yourself under real pressure, reverting to the person you were trying not to be. You were not failing because you lacked discipline or commitment. You were failing because you were working at the wrong level entirely. To understand why, you need to understand what confidence actually is, not the motivational definition, not the cultural one, but the biological one. Confidence, at its most precise, is the subjective experience of a nervous system that has enough resources, enough perceived safety, and enough internal coherence to move toward challenge rather than away from it. It is a state, not a trait. It is produced by specific physiological conditions, not chosen by a disciplined mind. And when those conditions are absent, no amount of positive thinking can manufacture the genuine article. What you get instead is performance. And performance always cracks under sufficient pressure because it is being held together by conscious effort rather than biological readiness. The neuroscientist Lisa Feldman Barrett has spent her career dismantling the intuitive model of emotion that most people, including most psychologists, operate with, the idea that emotions are detected by the brain, that the brain reads a situation, recognizes what is happening, and then generates the appropriate feeling. Her research reveals something far stranger and more consequential. The brain does not detect your emotional state. It predicts it, constantly, millisecond by millisecond. Your brain is running a simulation of what your body is likely to be experiencing, based on a continuous stream of physiological data arriving from every organ, muscle, and nerve ending you possess. What you experience as a feeling, including the feeling of confidence, is the brain's best guess about the state of the body, constructed from the inside out. This means that when your body is sending signals of depletion, threat, dysregulation, or exhaustion, your brain will construct an experience of insecurity, smallness, or dread, not because the situation warranted it, not because your thinking led you there, but because the body's data was unambiguous and the brain, always a faithful interpreter, render it faithfully into feeling. You cannot override this process by choosing better thoughts. Thought is downstream. It arrives after the prediction has already been made. This is the opening the door that Marsh's data revealed, and that this book intends to walk through completely. Because once you understand that confidence is a biological state rather than a psychological choice, the question changes entirely. It is no longer, how do I think more confidently? A question that sends you into an endless loop of self-monitoring and motivated reasoning that the very act of self-monitoring tends to destroy. The question becomes, what does my body need to feel safe enough, resourced enough, and coherent enough to produce the state I have been calling confidence? And that question has answers. Specific, biological, measurable answers that have nothing to do with mindset and everything to do with four interconnected systems inside your body that the confidence literature has never, in its entire history, treated as the primary terrain. The four systems of the autonomic nervous system, the endocrine system, the gut-brain axis, and the immune-inflammatory system. Each of these systems is directly involved in production and destruction of what we experience as confidence. Each of them is sensitive to inputs that have nothing to do with thought, light, movement, food, breath, touch, sound, sleep, social proximity, microbial ecology, hormonal rhythm. Each of them can be measurably shifted by interventions derived from their own biology, not from motivational logic. And each of them has been studied extensively by scientists who are not trying to write a confidence book, which is precisely why their findings have never made it into one. What Marsh found, in the end, was not a profile of a confident personality. She found a profile of a confident body, a body with high vagal tone and flexible autonomic regulation, with a testosterone to cortisol ratio that made challenge feel like opportunity rather than threat, with a microbiome diverse enough to produce the neurotransmitters that govern social ease and status comfort, with inflammatory markers low enough that the immune system was not quietly consuming the metabolic resources the brain needed to function at its most socially fluent. She found a body that had, through some combination of genetics and environment, and, critically, modifiable daily inputs, arrived at a state of biological readiness that the mind then experiences confidence and the world then perceived as charisma, competence, and calm authority. You are not reading the wrong thing. You have simply been given the wrong map. The territory you are trying to navigate is biological, and it has been there inside you, waiting for someone to hand you a map that actually matches the terrain. This book is that map. It begins not with your mind, not with your beliefs, not with your childhood stories or your limiting self-talk. It begins exactly where confidence itself begins, at the level of a nerve, a hormone, a microorganism, a single breath taken all the way to the bottom of the lungs. It begins with a body that has always known what your mind has spent decades trying to figure out. Chapter 2. The nervous system is the stage. Why your vagus nerve decides whether you perform or collapse before your brain gets a vote. In the winter of 1994, Stephen Porges was sitting with a dataset that refused to behave the way the textbook said it should. He was a neuroscientist at the University of Illinois, and he had spent years measuring heart rate patterns in newborns during surgical procedures, specifically tracking what happened to the autonomic nervous system under conditions of extreme physiological stress. What he kept finding was a response that the existing model of the autonomic nervous system could not adequately explain. The old model was elegantly simple. The autonomic nervous system had two branches, the sympathetic, which accelerated the body into fight or flight, and the parasympathetic, which slowed it back down into rest and recovery, activation and deactivation, gas and brake. This binary had governed neuroscience for a century. Porges's data did not fit it. There was a third response, a shutdown, a kind of biological freeze that was neither fight or flight nor rest and digest, but something older, stranger, and in some circumstances more dangerous than either. He followed that anomaly with particular obsession that occasionally produces genuine scientific revolutions, and what he eventually built from it, polyvagal theory, did not merely add a footnote to the existing model of the nervous system. It rewrote the architecture entirely, and buried inside the rewrite was something that no one had thought to look for in the nervous system before, a biological explanation for confidence. The vagus nerve is the longest cranial nerve in the human body. It travels from the brainstem down through the neck, into the chest, through the diaphragm and into the abdomen, touching the heart, the lungs, the stomach, the intestines, and most of the major organs along the way. It is a primary communication highway between the body and the brain, carrying far more information upward, from body to brain, than downward, a fact that most people find startling when they first encounter it, because we tend to imagine the brain as a sender and the body as a receiver. In reality, the body is talking constantly, urgently, in a language the brain has no choice but to listen to, and the vagus nerve is the primary cable through which that conversation travels. What Porges discovered is that the vagus nerve is not one structure but two, wrapped around each other across evolutionary time. The older branch, which we share with ancient vertebrates, governs the shutdown response, the freeze, the collapse, the biological playing dead that the nervous system deploys when threat is so overwhelming that fighting or fleeing is no longer viable. The newer branch, which evolved specifically in mammals and reaches its most sophisticated expression in human beings, governs something altogether different. The capacity for calm, engaged, socially open, curious presence in the world. Porges called the neural state produced by this newer branch the ventral vagal state, and when he began to describe what a human being in this state actually looks, sounds, and feels like, anyone who has ever been in a room with a truly confident person will recognize the description immediately. The voice is resonant and unhurried. The face is expressive and mobile. The eyes make contact in a way that feels welcoming rather than threatening. The posture is open. The attention is genuinely available. There is no performance of ease. There is actual ease produced not by a decision to relax, but by a nervous system that has received sufficient signals of safety to disengage its threat detection circuitry and allocate its resources toward connection and engagement rather than defense and survival. This is what confidence feels like from the inside when it is real. Not a thought, not a choice, a state, a full-body, measurable, physiological state produced by a specific circuit of the nervous system, available only when that circuit has been activated by the right inputs and unavailable regardless of intention or willpower when it has not. The implications of this are worth sitting with, because they are genuinely uncomfortable for anyone who has spent years believing that confidence is a matter of mental discipline. The ventral vagal circuit cannot be accessed through positive self-talk, because positive self-talk is a cortical activity, and the circuit Porges identified operates below the cortex, in the brainstem and the body, in territory that conscious thought does not govern. You cannot think your way into a ventral vagal state any more than you can think your way into a lower heart rate or a different blood pH. The circuit has its own inputs, its own language, its own triggers, and they're all physical, all sensory, all arriving from outside the world of deliberate cognition. What activates it, the research shows, is a perception of safety, not the intellectual conclusion that a situation is safe, but the body's direct sensory experience of safety signals. The sound of a warm, melodically varied human voice. The sight of a relaxed, open face. physical stillness in an environment without sudden threat cues, a breathing pattern that signals to the brainstem that the body is not in danger. These inputs are processed by what Porges calls a social engagement system, a network of nerves governing the muscles of the face, the middle ear, and the voice that evolved specifically to allow mammals to signal safety to one another and to receive those signals in return. When this system is online, the ventral vagal circuit opens, and with it comes everything we recognize as confident human presence. When it is offline, when the body has instead registered threat, uncertainty, or social danger, the sympathetic system takes over and the experience of confidence becomes neurologically inaccessible, not metaphorically but literally. In the same way that a light becomes inaccessible when the circuit powering it is broken, the measurable index of this capacity is called vagal tone, and it is one of the most important numbers almost no one outside of psychophysiology research has heard of. Vagal tone refers to the flexibility and responsiveness of the vagus nerve, specifically the degree to which heart rate varies rhythmically with the breath, a phenomenon called heart rate variability. When you inhale, the heart speeds slightly. When you exhale, it slows. The magnitude of that oscillation is a direct index of how responsive the vagal brake is, how quickly and fluidly the nervous system can shift between states. People with high vagal tone show consistently greater emotional regulation under stress, more flexible social behavior, higher tolerance for ambiguity and challenge, faster physiological recovery after disruption, and, critically, greater capacity for the kind of relaxed, engaged, risk-tolerant presence that the world reads as confidence. People with low vagal tone show the reverse across every measure. And vagal tone, unlike personality, is trainable. The first intervention that measurably increases vagal tone is so simple it seems almost offensive given the complexity of the science behind it. Extending the exhale. When the exhale is longer than the inhale, the vagal brake engages more deeply, the heart rate slows, and the brainstem receives a signal of safety that travels through the same neural channels as any other sensory safety input, which means it activates the ventral vagal circuit to the back door of the body's own respiratory rhythm, a ratio of roughly 4 counts on the inhale and 6 to 8 on the exhale. Practice for 10 minutes daily has been shown in multiple controlled studies to produce measurable increases in heart rate variability within 2 weeks. This is not relaxation advice dressed in scientific language. This is a direct intervention in the neural circuit that determines whether your nervous system reads the next high-stakes situation as a challenge to engage or a threat to escape. The second intervention is less intuitive and more socially radical. Co-regulation rather than self-regulation. The research of Jim Cohen at the University of Virginia on what he calls social baseline theory shows that the human nervous system did not evolve to regulate itself in isolation. It evolved to use the presence of other trusted bodies as its primary regulatory resource. When we are physically near people we feel safe with, our threat detection circuitry requires genuinely less energy to maintain the same level of vigilance, because the nervous system is distributing its perceptual load across multiple bodies the way a financial portfolio distributes risk. This means that the common advice given people struggling with confidence, spend more time alone, build your inner resilience, stop relying on others for validation, is, from a neurobiological standpoint, precisely backwards. The path to a more robust ventral vagal baseline runs directly through intentional, physical, genuine human co-presence, not away from it. The third intervention comes from Porges' own clinical work and from the research his theory has inspired. Deliberate immersion in acoustic environments that activate the social engagement system. The middle ear, governed by muscles connected to the vagal circuit, evolved specifically to filter the frequency range of the human voice out of background noise. And those muscles, like any muscles, can become slack through disuse. Environments dominated by low-frequency noise, traffic, machinery, the omnipresent hum of modern infrastructure actually suppress middle ear muscle tone and dampen the social engagement system's responsiveness. Environments rich in the melodic, variable frequencies of live human voices, music with dynamic range, or natural soundscapes do the opposite. This is not aesthetics. This is neurology. The nervous system you bring into a room full of people is shaped measurably by the acoustic history of the hours before you arrived. What Porges gave the world was not a therapy technique or a self-help framework. He gave a map of the biological substrate on which every confident human action is performed, and a precise account of why that substrate so often fails not because of weakness of character, but because of the specific sensory deprivations and threat cues that modern life delivers with such relentless consistency. The stage on which confidence performs is the nervous system. Before a single thought is generated, before a single word is spoken, before the conscious mind has formed any intention whatsoever, the nervous system has already decided which version of you is available to show up. The only question that matters, then, is what you are feeding it. Chapter 3. The Gut as Opinions. The microbiome research that rewrites the story of where confidence actually lives. In a laboratory at University College Cork in 2019, a team of researchers did something that should have made the front page of every newspaper in the world, and instead appeared quietly in a scientific journal, read by specialists, and passed almost entirely unnoticed by the culture that needed it most. They took gut bacteria from bold, exploratory rats, animals that have been behaviorally characterized as willing to enter open spaces, approach novelty, and tolerate uncertainty, and transplanted those bacteria into the intestines of timid, anxious rats, animals that hug walls, avoided open fields, and startled easily. Within weeks, the timid rats began to move differently. They entered open spaces. They approached unfamiliar objects. Their stress hormone profiles shifted. Their behavior, by every measurable index the researchers applied, had moved along the spectrum from avoidance toward exploration, from what, in a human being, we would recognize as self-protective withdrawal toward what we would recognize as confident engagement with an uncertain world. The bacteria had not changed their genetics. It had not changed their history. It had not given them better thoughts about themselves. It had changed the conversation happening between their gut and their brain. And that conversation, it turned out, was more determinative of their behavioral identity than anything the researchers had previously thought to measure. This result was not an accident, and it was not an anomaly. It was the inevitable destination of a scientific journey that had been building for 2 decades, as researchers in microbiology, neuroscience, psychiatry, and immunology began to discover, with mounting astonishment, that the approximately 38 trillion microorganisms living in the human gut were not passive residents performing housekeeping functions at the periphery of human biology. They were active participants in the production of mood, personality, stress reactivity, and social behavior, communicating with the brain through multiple channels simultaneously, influencing the synthesis of neurotransmitters, modulating the immune system's inflammatory output, regulating the vagus nerve sensitivity, and shaping, in ways that researchers are still fully mapping, the felt sense of who a person is in the world. The number that anchors everything else in this chapter is 90%. 90% of the body's serotonin is produced not in the brain, but in the gut, manufactured by specialized cells in the intestinal lining that are in constant biochemical dialogue with the microbial community surrounding them. This matters enormously for the story of confidence because serotonin is not, as popular culture has reduced it to, simply a happiness molecule. In the precise language of neuroscience, serotonin is a molecule of social rank. It governs a felt sense of status, belonging, and what researchers call social confidence, the background certainty that you have a legitimate place in the groups that matter to you, and that your presence there is welcome rather than merely tolerated. In studies of social hierarchies across species, individuals who occupy stable, secure positions in their social group show consistently higher serotonin activity than individuals in unstable or subordinate positions. What is less known, and far more important for our purposes, is that the relationship runs in both directions. Low serotonin function does not merely reflect low social confidence, it actively produces it, generating a kind of biological pessimism about social belonging that colors every interaction, every room entered, every conversation begun, with a faint but persistent undertone of anticipated rejection. If 90% of the serotonin shaping that experience is being made in the gut, and if the microbiome is one of the primary regulators of how efficiently that serotonin is synthesized and made available to the nervous system, then the question of gut health is not separate from the question of confidence. It is the same question, asked at a different level of the body. John Cryan and Ted Dinnan, the neuroscientists and psychiatrists who together have done more than anyone to build the clinical and scientific architecture of what they call the gut-brain axis, have spent years documenting specific bacterial species whose presence or absence produces measurable changes in social behavior, anxiety responses, and what they describe as proactive coping, the biological disposition to approach challenges actively rather than retreating from them, which is stripped of all cultural narrative, the behavioral signature of a confident organism. Lactobacillus rhamnosus, in research conducted by Cryan's group and replicated in multiple subsequent studies, was shown to produce changes in GABA receptor expression in the brains of mice that were comparable to the effects of anti-anxiety medications, and to do so through a mechanism that required an intact vagus nerve, meaning the bacteria was using the same neural highway that polyvagal theory identified as a foundation of the social engagement system to send its regulatory signals directly to the brain. When the vagus nerve was severed, the effect disappeared. The gut was not broadcasting into a void. It was in direct, specific, anatomically precise conversation with the nervous system that governs whether a human being feels safe enough to be fully present. And the microbiome was one of the primary authors of that conversation. What disrupts this conversation is not mysterious and not rare. It is the texture of ordinary modern life. Antibiotic use, even a single course, can reduce microbiome diversity by 30% and alter community composition for months or years. Ultra-processed foods, which now constitute the majority of calories consumed in many Western countries, have been shown to actively degrade the mucosal lining of the gut, the habitat in which the most beneficial bacterial species live, while simultaneously feeding the bacterial species associated with increased intestinal permeability, systemic inflammation, and the specific pattern of immune activation that researchers have linked to depressive and anxiety-like states. Chronic psychological stress, mediated through cortisol and the sympathetic nervous system, alters gut motility, reduces secretory IgA, and changes the selective pressures on microbial populations in ways that systematically favor stress-reactive bacterial communities over the calmer, more serotonin-supportive ones. The gut of a chronically stressed, sleep-deprived, ultra-processed food-eating modern human being is not a neutral biological environment for which confidence can be expected to emerge with any reliability. It is, at a microbial level, a confidence-hostile ecosystem, and no amount of mindset work takes place downstream of that reality without being colored by it. The sleep microbiome relationship deserves particular attention because it operates through a feedback loop that, once understood, reframes the entire cultural conversation about fatigue and performance. The microbiome regulates the production of several compounds that directly influence sleep architecture, including short-chain fatty acids that modulate the blood-brain barrier's permeability to tryptophan, the serotonin precursor, and certain strains of bifidobacterium that influence the circadian rhythmicity of cortisol secretion. Disrupted sleep, in turn, reduces microbial diversity, increases inflammatory bacterial species, and compromises the gut lining's integrity overnight, precisely the hours during which the most important microbiome repair processes are scheduled to occur. A person running on poor sleep is not merely tired. They are operating with progressively degrading biological confidence infrastructure, one that neither caffeine nor motivation nor cognitive reframing can compensate for. Because the problem is not in the mind that needs reframing. It is in the gut that is generating the neurochemical environment within which all thinking occurs. The practices that change this are not complicated, but they are specific, and their specificity matters because each one targets a distinct mechanism rather than a vague notion of wellness. Fermented foods, kefir, kimchi, sauerkraut, yogurt with live cultures, have been shown in a 2021 Stanford randomized controlled trial led by Justin Sonnenberg and Christopher Gardner to increase microbiome diversity and reduce inflammatory markers more effectively than a high-fiber diet alone over a 10-week period. The mechanism is direct inoculation of beneficial bacterial strains combined with the organic acids produced by fermentation, which shift gut pH in ways that favor the growth of GABA-producing and serotonin-supported communities. This is not a wellness trend. It is a confidence intervention at the level of neurotransmitter synthesis, and it begins to produce measurable neurochemical changes within weeks. Polyphenol, the compounds found in dark berries, green tea, extra virgin olive oil, dark chocolate, and the skins of deeply colored vegetables, function in the gut not as antioxidants in the simplistic popular sense, but as prebiotics, selectively feeding the bacterial species most associated with dopamine pathway integrity and reduced threat reactivity. The dopamine connection matters because dopamine is not merely a reward molecule. It is, at a systems level, the molecule of motivated approach, the biological signal that transforms a challenge from something to be survived into something to be pursued. The organism with abundant, well-regulated dopamine signaling is the organism that walks into uncertain situations with a forward lean of someone expecting something interesting rather than the backward lean of someone bracing for damage. And the microbiome is one of the upstream regulators of whether that signaling is robust or depleted. What the Cork experiment ultimately revealed, and what the entire field of nutritional psychiatry is now slowly, painstakingly confirming, is something that the confidence industry has never had the biological vocabulary to say. Some of what you've been calling your personality lives in your intestines. The shyness that arrives in rooms full of strangers. The reluctance that precedes every unrehearsed social encounter. The internal smallness that persists even when external circumstances argue loudly against it. Some of that is not psychology. Some of it is microbiology. And here's why this is the most hopeful sentence in this entire book. Unlike your childhood, unlike your genetics, unlike your accumulated history of social experiences that have calcified in memory into a story about who you are, your microbiome can be meaningfully transformed in 90 days. The community of organisms that is quietly co-authoring your experience of yourself is not fixed. It is a garden, and it responds to what you feed it with a fidelity that the mind, for all its complexity, has never quite managed to match. Chapter 4. Hormones are not your mood. They're your identity, and they can be renegotiated. In the early 2000s, a team of researchers began tracking the hormonal profiles of traders on the floor of a London financial trading firm, not during market crashes or moments of obvious drama, but across the ordinary art of trading days, measuring testosterone and cortisol in real time through saliva samples taken at specific intervals throughout the session. What they found demolished one of the most entrenched assumptions in the popular science of confidence, that hormones are responses to outcomes, chemical rewards, or punishments that the body distributes after the fact, like grades handed back after an exam. The data showed something far more dynamic and far more consequential. Testosterone rose not after successful trades, but before them, in anticipation of market engagement. And the magnitude of that anticipatory rise was a reliable predictor of the quality of decision-making that followed. More striking still was what the cortisol data revealed. It was not high testosterone alone that characterized the traders who performed with the most consistent confidence and clarity under pressure. It was a specific ratio, high testosterone relative to low cortisol, that distinguished them from their colleagues. And that ratio, the research showed, was not delivered by genetics or personality or years of experience alone. It was exquisitely, almost disturbingly sensitive to factors the traders were affecting every single day without knowing it. How they had slept, what light they had seen that morning, whether they had moved their bodies, and the subtle social dynamics of who had spoken to whom with what quality of regard in the hours before trading began. Robert Sapolsky, the Stanford neuroendocrinologist who has spent a career studying the relationship between hormones, hierarchy, and behavior across primate species, has documented with elegant precision how the hormonal architecture of an organism is not a fixed biological fact, but continuously updated social and environmental negotiation. In baboon troops he observed over decades in Kenya, the testosterone to cortisol ratio of dominant males was not primarily a cause of their status. It was a dynamic reflection of the stability of that status, rising when their position was secure and supported by social alliances, falling when it was threatened or isolated. The hormones were reading the social environment and generating the biological conditions for behavior appropriate to it, which means that the environment was, in a very real sense, writing the hormonal script that the animal then performed as personality. The baboon did not decide to be confident. The baboon's endocrine system produced a state that expressed itself as confidence because the conditions, social, environmental, physical, supported that production. The human endocrine system operates by the same logic, with the same sensitivity to context, and with the same radical indifference to what the conscious mind would prefer. Testosterone, which exists in meaningful quantities in all bodies regardless of biological sex, and which functions in both as a primary driver of approach motivation, risk tolerance, and a felt sense of social potency, is not a stable hormonal identity. It fluctuates across a range that can vary by 300% or more depending on sleep quality, physical activity, social experience, light exposure, and perceived safety or threat of the social environment. Cortisol, the stress hormone that in chronic elevation suppresses testosterone production, degrades hippocampal function, narrows attentional focus on the threat, and produces a biological state that the mind experiences as inadequacy, self-doubt, and a persistent sense that the room is more dangerous than it looks, is equally dynamic, shaped hour by inputs that the person generating it is often entirely unaware of managing. The consequence of this dynamic interplay is that what feels like a stable identity, I am someone who shrinks in groups, I am someone who cannot project authority, I am someone for whom confidence is always effortful and always temporary, may in significant part be a hormonal pattern that the body has settled into through accumulated environmental conditioning, not a fixed self that was assembled once and cannot be renegotiated. The ratio that determines whether a challenge registers in the body as an exciting opportunity or a threatening exposure is not set in stone. It is set in biology, which means it is set in the same biological medium that sleep, light, movement, and connection continuously and measurably reshape. The science of Sari Van Anders, whose work on testosterone and social behavior has repeatedly complicated the crude pop science narrative of the hormone as simply a dominance molecule. Shows that testosterone responds not to the fact of winning, but to the quality of the engagement, specifically to experiences of what she calls confident social agency, the feeling of acting from genuine motivation in social context rather than performing compliance or managing threat. This distinction is critical because it means that simply placing yourself in high-status situations does not reliably raise testosterone. In fact, situations where you feel overexposed, under-resourced, or socially unsupported can suppress it sharply by elevating cortisol simultaneously, creating precisely the unfavorable ratio that makes confident behavior harder to produce. The hormonal environment follows the quality of the social and physical experience, not the external markers of its prestige. Oxytocin enters this picture as a molecule that has been systematically mischaracterized in popular science as a simple bonding or love hormone, and in doing so has had its most important function for the story of confidence almost entirely overlooked. The research of Sue Carter, Kirsten Ovnes Moberg, and others working in the oxytocin literature shows that the hormone's primary function in the social nervous system is threat reduction. Specifically, it suppresses amygdala reactivity to social threat cues, reduces the cortisol response to uncertainty, and lowers the threshold at which the nervous system is willing to classify an unfamiliar situation as safe enough to engage with openly rather than guardedly. In practical terms, oxytocin does not make you feel more loving. It makes the world feel less dangerous. It is a biological confidence amplifier that works by narrowing the gap between what the nervous system registers as threat and what it registers as opportunity. And it is released not by achievement or status, but by the specific quality of genuine, trusting, physically present human connection. This is why isolation consistently degrades confident behavior across species, and why the advice to develop inner resilience by spending more time alone, common in productivity culture and certain strands of self-help philosophy, tends to produce not confidence, but a more defended version of the same anxiety, one that has learned to perform self-sufficiency, while the hormonal substrate of genuine social ease quietly erodes. The oxytocin system needs regular activation through real connection to maintain the suppression of threat reactivity that makes confident behavior biologically accessible. Without that activation, the amygdala's default setting, vigilant, cautious, prone to interpreting ambiguous social cues as negative, gradually reasserts itself, and confidence becomes something that requires more and more effortful performance to approximate. Morning light, arriving in the eyes within 30 minutes of waking, triggers a cortisol awakening response, a deliberate, evolutionarily designed spike in cortisol that serves not as a stress signal, but as a biological activation sequence, priming the system for alert engagement with the day's challenges and establishing the diurnal rhythm from which subsequent hormonal patterns unfold. When morning light is absent, when the first light the eyes receive is the blue-shifted glow of a phone screen in a dim room, this awakening response is blunted, the circadian anchor is weakened, and the downstream effects ripple through testosterone production, melatonin timing, and the overall hormonal coherence of the day in ways that measurably reduce the biological resources available for the kind of confident, forward-oriented engagement that the day will inevitably demand. 10 minutes of outdoor light before any screen contact in the morning is not a wellness recommendation. It is an endocrinological intervention with a documented mechanistic pathway, resistance exercise, specifically the kind that loads large muscle groups through compound movements at sufficient intensity to generate genuine metabolic demand, produces a testosterone to cortisol ratio most associated with confident challenge-seeking behavior through a mechanism distinct from any other form of physical activity. The acute cortisol rise that accompanies intense resistance training is followed, in the hours and days after training, by a compensatory testosterone elevation and cortisol suppression that shifts the ratio in the direction associated with approach motivation, reduced threat sensitivity, and the biological disposition to enter difficult situations with expectation rather than dread. This effect is not produced equally by all exercise. Prolonged endurance activity at high intensity chronically elevates cortisol without the compensating testosterone response and can actually degrade the ratio over time. The specificity matters because it means the biological return on physical effort depends not on the effort itself, but on its type, duration, and recovery structure. What endocrinology ultimately reveals about confidence is something that neither psychology nor motivational philosophy has ever been equipped to say with this kind of precision. The felt sense of who you are in a challenging situation is being continuously written by hormones that are themselves being continuously written by your sleep, your light exposure, your movement, your social environment, and the quality of your most intimate connections. Identity at this level of analysis is not a narrative. It is a chemical state. And chemical states, unlike narratives, have inputs that can be identified, measured, and changed. The person who feels chronically unable to project confidence is not suffering from a character deficiency. They are running a hormonal pattern that their daily environment has been reliably reproducing, and that pattern is as renegotiable as the environment that generates it, which is to say, entirely. Chapter 5. The Ancestral Body. What evolutionary biology reveals about why confidence was never meant to be a personal achievement. There is a village in the western rift of the Hadza territory in northern Tanzania where, on certain nights, the fire burns until the sky begins to pale and the dancing does not stop until it does. Christopher Bohm, the evolutionary anthropologist who has spent more of his professional life inside hunter-gatherer societies than perhaps any living researcher, has sat beside fires like this one and watched what happens to people around them over the course of hours, how the drumming reorganizes breathing across the group into something approaching synchrony, how bodies that arrive at the fire carrying the residue of the day's uncertainties gradually shed it not through conversation or reflection but through shared rhythm, shared heat, shared physical proximity in the dark. He was not watching a cultural ritual. He was watching a biological technology, one that the human nervous system had been shaped by several 100,000 years of selection pressure to require, and one that the modern world had, in the span of a few industrial generations, almost completely dismantled and replaced with nothing that the body recognized as equivalent. Bohm's broader research into the social architecture of confidence in hunter-gatherer societies produced findings that should permanently alter how anyone thinks about the confidence problem in modern life. Across the societies he studied, the Hadza of Tanzania, the Kungsan of the Kalahari, the Mbiti of the Congo Basin, he found that what Western psychology would classify as confident behavior was not primarily generated by individuals working on their inner states. It was produced and regulated by the group, distributed through the social body via specific collective practices, and maintained to the kind of continuous physical co-presence that modern life has reclassified as unnecessary or even as a failure of self-sufficiency. Status in these societies was not a ladder to be climbed through individual performance. It was a fluid, collectively negotiated social ecology in which the confidence of each member was partly a function of the group's active investment in maintaining it. Bohm called this dynamic reverse dominance hierarchy, the systematic suppression of individual status seeking in favor of collective stability. And what is remarkable about it from a neurobiological standpoint is that it produces, as a social byproduct, precisely the oxytocin-rich, low cortisol, high serotonin hormonal environment in which confident behavior is most biologically accessible for the greatest number of people simultaneously. The human nervous system was calibrated in environments like this one, not in offices, not in cities, not in the atomized social arrangements of modernity, where the average person maintains genuine physical intimacy with a shrinking handful of people and spends increasing proportions of waking life in a company of screens that deliver social information without delivering the physiological inputs the nervous system uses to determine whether that social information should be classified as genuine belonging or as its pallid digital simulation. The gap between the social environment the body evolved to inhabit and the social environment it actually occupies in the 21st century is not a cultural inconvenience. It is a biological emergency unfolding in slow motion, and one of its most consistent casualties is a capacity for effortless, confident presence. The state that, in the ancestral environment, was a default output of a nervous system adequately supplied with the inputs it was designed to receive. The research on interoception, the body's capacity to perceive its own internal physiological state, illuminates a particular dimension of this mismatch that almost no one in the confidence literature has thought to examine. Interoception is mediated primarily through the vagus nerve and the insular cortex, and it governs the accuracy with which a person can read their own body signals: heartbeat, gut sensation, breath quality, muscular tension. The subtle proprioceptive data that tells the organism below the threshold of conscious thought whether it is safe, resource, and ready to engage, or depleted, threatened, and in need of withdrawal. High interoceptive accuracy has been consistently associated in the research with better emotional regulation, more flexible social behavior, faster recovery from stress, and, in what is becoming one of the more robustly replicated findings in embodied cognition, more reliable access to the physical sensations of confidence that the brain needs to construct the psychological experience of it. The brain, remember, predicts rather than detects, and it builds those predictions from the body's data. A person who cannot accurately read their own body's data is a person whose brain is building its predictions from noise, generating emotional states that bear an uncertain relationship to the organism's actual condition. Sedentary, screen-dominated modern life degrades interoceptive accuracy through a mechanism that is straightforward once understood. Interoception requires movement to generate the proprioceptive and cardiovascular signals that the insular cortex uses to build its model of the body's internal state. A body that sits for most of its waking hours, that rarely experiences the full range of its cardiovascular capacity, that navigates the world primarily through the thin sensory channel of eyes and fingertips, is a body that is progressively losing its ability to sense itself, to feel its own aliveness from the inside, and with that loss goes much of the visceral, physical confidence signal that the brain would otherwise have available to construct the experience of confident presence from. This is not a metaphor about being disconnected from yourself. It is a literal description of insular cortex input deprivation, and its effects on confidence are as real and as measurable as the effects of sleep deprivation on executive function. What the ancestral body used to maintain both interoceptive richness and the social hormonal environment of confident belonging was a suite of practices that modern life has not improved upon so much as abandoned. Shared physical challenge, the coordinated exertion of hunting, building, carrying, climbing, produce simultaneous testosterone elevation, oxytocin release through physical co-presence and synchronized effort, endorphin response through sustained exertion, and a particular quality of nervous system coherence that comes from bodies moving together toward a shared goal in physical space. The neurological aftermath of such activity was not mere tiredness. It was a deep recalibration of the threat detection circuitry, a kind of biological reset in which cortisol cleared, vagal tone elevated, and the social safety assessment that the ventral vagal system runs continuously was updated with fresh evidence that the group was intact, the bonds were real, and the world was navigable. What the modern gym, for all its genuine physiological benefits, almost never replicates is the social and rhythmic dimension of this reset, the fact that it was done with others in synchrony, with a shared stake in the outcome. The biology of the activity was inseparable from the sociology of it, and extracting the physical component while discarding the social one produces a measurably different neurobiological outcome. Rhythm deserves particular attention because its effects on the nervous system are among the most robustly documented and most consistently ignored by any literature not explicitly engaged with music therapy or somatic psychology. Entrainment, the tendency of biological oscillators to synchronize with external rhythmic stimuli, is not a poetic metaphor for being moved by music. It is a measurable neurophysiological phenomenon in which heart rate, breathing, neural oscillations, and the hormonal pulses that regulate arousal and social readiness begin to align with rhythmic auditory and tactile input, drumming, chanting, and rhythmic movement. Present in every human culture for which records exist are not decorative expressions of culture. They are biological technologies for producing rapid, reliable nervous system coherence across large groups simultaneously. The confidence that emerges from collective rhythmic practice is not a psychological attitude generated by positive social experience. It is a physiological state produced by synchronized neural oscillation and its downstream effects on vagal tone, oxytocin release, and the suppression of the default mode network's tendency towards self-referential threat processing, toward the relentless internal narration of inadequacy that most people experience as the opposite of confidence. Touch, too, belongs in this accounting, not as a soft consideration, but as a hard biological input. Cutaneous pressure activates the C-tactile afferent nerve fibers that project directly to the insular cortex and trigger oxytocin, released from the hypothalamus, a pathway so direct and so powerful that its consistent absence in social environments produces measurable elevations in inflammatory markers and cortisol over time. The progressive reduction of casual, affectionate physical contact in many modern Western social environments, driven partly by cultural anxiety about touch and partly by the replacement of in-person interaction with digital communication, is not a neutral social evolution. It is a removal of a biological input that the nervous system uses to regulate its threat assessment of the social world, and its removal leaves the system in a state of chronic mild social alarm that is functionally indistinguishable at the level of hormonal and neural effects from the low-grade anxiety that most people simply accept as their baseline. Eye contact, the last of the ancestral inputs worth examining in detail, activates the social engagement system through the superior temporal sulcus and triggers a cascade of neural and hormonal events, including oxytocin release, pupillary dilation associated with approach motivation, and the rapid, below-conscious mutual assessment of social safety that the ventral vagal circuit depends on that no digital equivalent has yet managed to replicate. Video calls deliver the image of eyes but not the neurological event of mutual gaze, because mutual gaze requires the simultaneous activation of both participants' superior temporal sulci through actual shared visual space, a condition that the geometry of camera placement and screen viewing makes neurologically impossible. The confidence that flows from feeling genuinely seen by another person in physical space is not a feeling that can be adequately approximated by any technology currently available because it is generated by a biological event, not a visual one. And the biology requires bodies in proximity to occur. What Bohm found at those fires in Tanzania, and what the entire literature of evolutionary mismatch converges on, is a picture confidence not as a personal achievement to be constructed in isolation through discipline and mindset work, but as a collective biological state that the human organism was designed to produce in community through physical co-presence, shared challenge, rhythm, touch, and the specific neurological safety of belonging to a group of people who know your name and your body and your history. The modern project of building confidence alone in front of a mirror through self-talk and visualization and individual habit stacking is not merely ineffective, it is, at the deepest evolutionary level, a category error. It is asking the body to generate, through solitary internal effort, a state that its entire biological architecture was built to produce together. Chapter 6. Becoming the Signal. How to make your biology produce confidence as a default state rather than an occasional miracle. In 2017, a neuroscientist named Dr. Martin Paulus, research director at the Navarro Institute and formerly at the University of California, San Diego, published the findings of a 20-year study into the neurobiology of elite performance under pressure, drawing on data from Navy SEALs, Olympic athletes, emergency room surgeons, professional musicians performing before thousands, and entrepreneurs navigating the specific terror of consequential public failure. He had expected, going in, to find a neurological profile of fearlessness, brains that simply registered less threat, nervous systems that were constitutionally calmer, endocrine systems that released less cortisol in the face of challenge. What he found instead was something that required him to rewrite the central question of his research entirely. The people who performed with the most consistent confidence across the widest range of high-stakes situations were not the people who felt the least disrupted by challenge. They were the people whose bodies recovered fastest from the disruption that challenge always, without exception, produced. The difference between the elite performer and the person who collapses under pressure was not the presence or absence of the biological stress response. It was the speed and completeness of the return to coherent functioning after that response had done its work. This finding, deceptively simple, neurologically profound, reframes the entire project of building confidence at a biological level because it shifts the target from a state to be permanently inhabited to a capacity to be systematically strengthened. No one, including the most apparently confident person in any room, operates in an unbroken ventral vagal state. No one maintains a perfectly favorable testosterone to cortisol ratio across every moment of every day. No one's microbiome produces serotonin with perfect consistency regardless of what it is fed. The biological systems that generate what we experience as confidence are dynamic, responsive, and perpetually subject to disruption by the ordinary demands of human life. And that is not a flaw in the design. It is a design. The question was never how to eliminate disruption. It was always how to build a body that moves through disruption the way water moves through rock, shaped by it temporarily, restored to itself inevitably, and subtly more capable with each passage. The measurable index of this capacity is heart rate variability, and in the population of elite performers Paulus studied, the HRV profiles were distinctive in a way that correlated with everything else the book has established. High resting HRV indicated a vagus nerve that was responsive, flexible, and capable of rapidly deploying the ventral vagal brake after sympathetic activation, a nervous system, in other words, that could enter the threat state that challenge demands and exit it cleanly rather than remaining trapped in the arousal long after the challenge had passed. The trapped arousal, sympathetic activation that lingers, cortisol that stays elevated, a nervous system that cannot find its way back to the ventral vagal baseline, is a biological substrate of what most people experience as a confidence problem. It is not that the stress response fires. It is that it does not complete. The body mobilizes for threat and then cannot fully demobilize, leaving the person in a state of chronic partial activation that colors every subsequent situation with residual threat, narrows the attentional field, suppresses the social engagement system, and makes the construction of confident presence neurologically expensive in a way it was never meant to be. Consider a woman named Sarah, a documentary subject in a longitudinal study at the University of Amsterdam tracking biological markers of performance psychology across 3 years. She arrived at the study self-described as someone who had struggled with confidence her entire adult life, a writer and editor who could perform competence fluently in most professional situations, but who experienced what she called a persistent internal gap between how she appeared to others and what she felt herself to be, a gap that widened dramatically under conditions of public visibility or genuine creative risk. Her baseline measurements were consistent with everything the research predicts, resting HRV in the lower quartile for her age group, a blunted cortisol awakening response suggesting a dysregulated diurnal cortisol rhythm, microbiome diversity below the median with notably low levels of lactobacillus and bifidobacterium species, inflammatory cytokine levels elevated in the range associated with subclinical depressive symptoms, and interoceptive accuracy scores that placed her in the lower third of the study cohort, meaning that her ability to accurately read her own body's internal signals was significantly compromised. She was not, as she had always assumed, a person with a confidence problem. She was a person with biology that had been consistently generating the internal conditions for self-doubt and threat sensitivity, and she had spent years trying to manage the psychological output of those conditions without ever touching conditions themselves. Over the following 18 months, Sarah made a sequence of changes that were biological rather than psychological in their logic. Changes derived not from a motivational framework, but from the specific mechanisms the research identified as most modifiable and most directly implicated in the production of her particular biological profile. She began with the exhale extended breathing protocol, 10 minutes each morning, because the vagus nerve was the most acutely dysregulated system and the one with the most immediate and measurable response to direct input. Within 3 weeks, her resting HRV had shifted measurably upward. Within 6 weeks, she reported, in language that precisely mirrored the polyvagal literature without her having read it, that social situations felt physically different, that the room arrived at her body as less immediately threatening, that she had more time, more spaciousness between stimulus and response. She was not thinking differently about social situations. Her nervous system was processing them differently because the biological substrate of that processing had changed. She added morning light within the first month, outdoor exposure within 30 minutes of waking before screen contact, and tracked the change in her cortisol awakening response through the study's salivary cortisol monitoring. The blunted morning cortisol that had characterized her baseline gradually resolved into a more robust awakening peak over 8 weeks, and with it came a quality of morning alertness and forward orientation that she described as feeling, for the first time, like her body was on her side in the early hours rather than working against her. The testosterone to cortisol ratio, tracked through the study's monthly hormonal assays, began shifting in a direction associated with challenge-seeking rather than threat avoidance. She had not changed her thoughts about mornings. She had changed the light information her hypothalamus received at the moment of the day, when the entire hormonal cascade of the next 16 hours was being set. The dietary changes came third, and they came slowly, not as a dietary overhaul, but as a targeted microbiome intervention, daily fermented food, polyphenol-rich vegetables, a reduction in the ultra-processed foods that have been degrading her gut lining and suppressing her bifidobacterium populations. At the 6-month mark, microbiome sequencing showed measurable increases in diversity and in the specific bacterial species most associated with GABA production and reduced social anxiety. She reported less of the low-grade social dread that had preceded group situations for as long as she could remember, not its complete absence, but a reduction in its baseline intensity that made it feel, for the first time, manageable rather than defining. The gut had changed. The conversation between gut and brain had changed. The neurochemical environment in which she was generating her experience of herself in social space had changed. Resistance training entered at month 4. 2 sessions per week of compound movement at genuine intensity, with full recovery between sessions, not for fitness, but for the specific hormonal ratio the research identified as the biological signature of confident challenge seeking. The testosterone elevation and cortisol suppression that followed each training cycle produced, in the days after each session, a measurable shift in her threat perception threshold, a window of biological confidence readiness during which she began deliberately scheduling the situations that had previously felt most exposing. She was not using willpower to enter difficult situations. She was timing her entries to coincide with the hormonal conditions her body had generated, making confident action biologically easier rather than psychologically heroic. The social restructuring was the last and in some ways the most personally demanding change, not because it required the most effort, but because it required the most honesty about what the research was saying. She reduced her time on social media platforms that delivered social information without social biology and increased the frequency and quality of in-person time with a small group of people with whom genuine safety existed. She initiated one shared physical activity with this group each week. Nothing elaborate, a long walk, a group cooking session, a collaborative physical project. Not because she had read about ancestral confidence practices, but because the research on social baseline theory had made it unmistakably clear that her nervous system was using the physical presence of trusted others as a regulatory resource, and that she had been systematically depriving it of that resource while wondering why her self-regulation in social situations felt so effortful. At 18 months, Sarah's biological profile was unrecognizable by the study's measures. Resting HRV in the upper quartile, cortisol awakening response robust and well-timed, microbiome diversity above the study median, inflammatory markers normalized, interoceptive accuracy in the upper third. She had learned, through the accumulation of bodily experience generated by movement, breath, and social co-presence, to read her own internal signals with a precision that now gave her real-time access to the difference between genuine threat and the residual noise of a nervous system that was no longer chronically dysregulated. She did not report feeling fearless. She reported that fear, when it arose, felt like information rather than verdict, a signal the body was sending about the significance of the situation rather than a pronouncement about her adequacy to meet it. This is a distinction that Paulus's research identified as the defining neurological difference between the people who perform confidently and the people who do not. Not the absence of the disruption signal, the speed and completeness of the return from it. The biology of confidence, assembled across the 6 chapters of this book, is not a complicated picture once it is seen whole. The nervous system needs safety signals, breath, sound, social presence, co-regulation, to access the ventral vagal state that makes confident engagement biologically possible. The endocrine system needs light, movement, connection, and sleep to maintain the hormonal ratio that makes challenge feel like opportunity rather than threat. The gut needs microbial diversity, fermented food, polyphenol richness, and sleep quality to sustain the neurotransmitter production that generates the social confidence, dopamine motivation, and threat suppression that the brain uses to construct the felt sense of being adequate to the moment. The ancestral body needs physical co-presence, shared challenge, rhythm, and touch to receive the biological inputs that the nervous system evolved to require for production of confident belonging. And the whole integrated system needs, above all, the capacity for fast recovery from disruption, the biological resilience that is not the absence of the stress response, but the training of every system to return to coherence after it. None of this is mindset. None of this is habit in the motivational sense. None of this requires the reader to believe something about themselves that they do not currently believe, or to perform a version of themselves that they have not yet become. It requires only the recognition that the body is not the obstacle between you and confidence. It is the instrument through which confidence is produced, and it responds to biological inputs with fidelity and a speed that no amount of psychological intervention has ever reliably matched. The first lever pulls the one closest to brainstem. The breath, the exhale, the 10 minutes of extended expiration that tells the oldest part of the nervous system, in the only language it has ever understood, that the threat is past, the body is safe, and the full resources of a coherent human organism are available right now, for whatever the moment requires. The signal that it is working is not a thought. It is a sensation, a quality of physical spaciousness in the chest, a slight drop in the shoulders, a change in the way the room arrives to the senses, as though the aperture through which you are perceiving the world is quietly, measurably widened. That is the ventral vagal circuit opening. That is the body producing the state that the mind will, a moment later, experience as confidence, not as a miracle, not as an accident, but as a reliable output of biology that has finally been given what it needed to do what it was always capable of doing.